Thank you very much, and it's a real pleasure to be able to speak at this particular event and revel in the amazing innovations that all of my colleagues are generating here in the MGB system. So I want to take you on a journey into another cell type. You hear a lot of different technologies, but to the B cell, and why we're working on it. So there's a significant unmet need for a cell therapy in the context of regenerative medicine, and despite 40 years of development, there are no FDA-approved cell therapies in the context of regenerative medicine. And the problems are principally that allogeneic therapies face the challenge of immune rejection, and then autologous treatments are expensive and challenging to scale. Well, biology has delivered a solution to us, which is not uncommon. We have cells which are capable of regenerative capacity, but this one was surprising, and we have patents now to support this and research work. We discovered that the allogeneic naive B cell has both anti-inflammatory, immune modulatory, and tissue regenerative functions. This is exciting. The B cell is known, as you know, for making, it's an incredible factory for making antibodies and doing some antigen presentation on the side. But in addition to that, this remarkable cell is capable of this, and in the context of neural injury, is both neuroprotective and neuroregenerative. This was surprising data. These cells are readily accessible in your bloodstream. They're safe. We've demonstrated they can be locally or intravenously administered and only survive for 10 to 14 days, but effect long-term therapeutic effects. And there's no immune rejection that we've observed in either local or systemic delivery of those allogeneic naive B cells. We've demonstrated the mechanism of action of these cells, which involves their interaction with other immune and inflammatory cells in the body, and that includes microglia in the brain, macrophages in the blood, and in the tissues. So our data is fascinating in the sense that we've demonstrated in multiple models, validated preclinical models of diabetic skin wounds, traumatic brain injury, and most recently ALS, all published work, significant impacts in healing when, let's say in the case of skin wound or TBI or traumatic brain injury, we locally deliver allogeneic naive B cells to the site, causing significant acceleration in wound healing by 6 days in a skin wound, decreasing lesion size by 35% in the context of TBI, and finally leading to very significant delays in disease onset and progression by 28 days in the context of a highly reproducible model of ALS. Our first clinical indication that we chose was intracranial hemorrhage. This is a deadly and disabling disease with no effective therapies currently out there. 20 to 30% mortality, almost half of the survivors suffer long-term morbidity, and obviously a very substantial cost burden due to that morbidity in the long term. And here we propose local delivery of the allogeneic naive B cells. This would be an off-the-shelf product in this context. Our data to date that has been gathered at the moment shows a significant impact of the B cell therapy delivered 24 hours after intracranial hemorrhage using a number of different neurological studies. This is the Rotarod, which measures balance, coordination, and motor planning. And here in red is the intracranial hemorrhage plus the allogeneic naive B cells. In the blue line below, the intracranial hemorrhage without therapy, demonstrating a functional recovery of 20% for 14 days following the therapy. Now, in addition, we're very keenly aware that everything has to be clinically applicable. And so it's all very well demonstrating all of this stuff in mice. You have to develop a GMP allogeneic B cell isolation process, in our case, which we're developing, that's proprietary and patentable. and doing formulation stability and storage studies, which have been completed with a very, very experienced team at the Dana-Farber Cancer Institute Stem Cell Therapy Center. And we can convert this process into full GMP, do cell expansion studies, and generate now with potentially proprietary methods enhanced immune regulatory and neuroregenerative cells in that context. The commercialized opportunity here is, we think, significant. This is a broadly applicable technology. Again, the product itself is a off-the-shelf allogeneic naive B cell that's readily accessible from the leukapheresis of patients or healthy individuals, and has what we think a high-impact lead indication of intracranial hemorrhage. And we currently have an ongoing interact going with the FDA on that, with preclinical proof of concept and obviously a pipeline into first in human. The second thing is our follow-up indication, which has been funded extensively by private foundations, is in ALS. And we have a pre-IND in place for October of this year to discuss a phase 1 trial for the intravenous delivery of allogeneic naive B cells in the context of patients with ALS. And I would highlight the point that we've already demonstrated safety and feasibility of this approach in 2 patients in the context of a compassionate use FDA approval. We have multiple patents granted and filed on applications of the allogeneic naive B cells, and underway proprietary methods for preparation and manipulation in that process. We have a GMP cell isolation process from donor leukapheresis to that product, and a clear pathway, given our amazing collaborators, to a GMP process. We've demonstrated stability and potency of our product to date over 6 months in liquid nitrogen storage. So why am I here? I'm here, why all of the speakers here. We want to take our innovations to serious first-in-human studies in the context of phase 1 trials. We've generated $1 million in angel funding to get our company Celebrium started, and we've raised many more millions than that in non-dilutive funding from government and private foundations to make this technology move forward. However, now we're at that advanced preclinical development IND stage and to the completion of a clinical trial where we come to investors for their vision and their funding to make this possible. Thank you very much indeed.